Good day everyone. Today I'm going to present Penguin, a tool for predicting pseudouridine sites in Nanopore direct RNA sequencing data. And this is a joint work with Daniel Acevedo, Swapna Vidor, Husina Mir, and Saras Janka. As outlines for this presentation, I will start by describing the motivation, the problem statement, and the existing solution. Then I will focus on our main contribution and our main approach pipeline and workflow. Then I will describe how we generate the benchmark datasets. Then I will show the performance evaluation results and the abundance of detected pseudouridine sites as well as its extent. Finally, I will conclude with some directions for future work. As a motivation for this work, pseudouridine is one of the abundant RNA modifications that occurs at the uridine site and catalyzed by pseudouridine synthase. It plays an important role in many biological processes such as stabilizing RNA through enhancing the function of transfer RNA and ribosomal RNA. The modification of uridine to pseudouridine has been observed in nearly all kinds of RNA, including but not limited to transfer RNA and messenger RNA. Pseudouridine has also important roles in drug development and response to stresses. The problem in identifying pseudouridine sites in RNA modifications comes from that some experimental technologies that have been developed to detect pseudouridine sites are both time-consuming and expensive. Therefore, there was a direction in the literature to address this problem using computational biology methods to identify pseudouridine sites. As for the main contribution of this work, the third generation sequencing technology platform provided by Oxford Nanopore technology has been proposed as a new means to detect RNA modifications on long-read RNA sequence data. However, and to our knowledge, this technology has not been used to identify pseudouridine sites in RNA sequence data. To this end, our work aims to address this direction by developing machine learning models integrated into a tool called Penguin to identify pseudouridine sites in direct RNA nanopore sequencing data. The main pipeline of Penguin is shown in this slide, and as we see, the pipeline starts by extracting cells from an animal in the cell's culturing phase. Then we extract the RNA from the cells during the library preparation phase, and after that we put the RNA through the Nanopore mini-ion device to start generating signal data. Next, FAST5 file that is produced by the mini-ion device and store the output of that device in form of electrical signals that comes with that device, in addition to a SAM file that is generated by aligning the RNA sequence reads to reference genome are input to Penguin platform using the produced SAM file and a provided bit file which highlight the target pseudouridine modification locations. A coordinate file is created with the IDs of the files that have the target modification. Next, Nanopolish software is launched by our Penguin platform for performing signal extraction. The output of this stage is a dataset of signal samples. Next, the samples that are related to pseudouridine target modification are filtered from the samples generated from the signal extraction. Next, and using the information in the coordinate file, some of the filtered samples will be labeled as modified with pseudouridine, and the remaining will be labeled as unmodified. Next, some features will be extracted from those modified and unmodified samples, which are fed to the machine learning classifiers in the training phase. Using those features, the classifier will be able to predict the pseudouridine site in the testing phase for different cell lines. The extracted features include the length of the signal, the mean, the standard deviation, and the one-hot encoding of the reference schema, which is produced by aligning the RNA sequence reads to a reference genome. Currently, Penguin platform integrates several developed machine learning models, including to identify pseudouridine sites in Nanopore direct RNA sequencing data, includes support vector machine, random forest, and neural network. As for generating the benchmark dataset, two different benchmark datasets were generated, one for HAK and another for HeLa cell lines. The former is used for training the machine learning classifier of Penguin, while the latter was used for testing the machine learning classifier. As for the performance evaluation results, we randomly divide the benchmark dataset of HAK cell line into two folds, one for training and another one for testing. The test size was set for 2.2, which means 80% of benchmark dataset is used for training the model and 20% of the dataset is kept for testing the model. And in this case, we do performance evaluation using the same cell line, and support vector machine performs the best, then neural network, then random forest. And those are the learning curve for the classifier. Also the flow chart D show part chart for the classifier that show their precision recall AUC. Here also are the ROC curve for each classifier. Next, we did performance evaluation for feature importance, or to figure out the feature importance. And in this case, we built 5 versions of the each Penguin machine learning applied model to HAK benchmark dataset. In the first 4 versions, each developed model is trained with one of the 4 extracted features, while in the 5th version, each model is trained with the combination of all 4 features. Also, we did performance evaluation against independent cell line. In this approach, we used HAK cell line benchmark dataset for training a machine learning model, and HeLa cell line dataset for testing their performances. Penguin was able to detect the abundance of pseudouridine sites, and in order to identify such abundance in RNA sequence of either HAK or HeLa cell line, we first ran the best machine learning model in Penguin, which is SVM, on the unlabeled benchmark dataset of HAK and HeLa cell line. Then we identify all humor samples predicted as pseudouridine ones in both datasets. Next, we can identify the number of pseudouridine unique genomic location as well as their frequencies for the 2 complete cell lines. Here we found that there are around 556 unique genomic location of pseudouridine in HAK. Similarly, that there are around 39 unique genomic location of pseudouridine in HeLa cell line. As for the overlapping between unique genomic location of pseudouridine in both cell lines, we found around 6,482 unique genomic location of pseudouridine that are common between both cell lines. Also, we found that there is an overlapping of 15.8 between the top first percent frequent modified pseudouridine genes of both complete HeLa and HAK cell lines. Penguin also was able to detect the extent of pseudouridine modification. We noticed that the extent of pseudouridine modification identified by the number of humor samples predicted as pseudouridine samples to the total number of humor samples in the complete RNA sequence of HAK cell line is much greater than in its counterpart for HeLa cell line. Actually, it was 9% for HAK versus 0.5 for HeLa cell line. In conclusion, we found Penguin platform outperforms existing prediction models of pseudouridine sites in RNA sequence in the literature that achieved an accuracy of 76% at most when testing with independent cell line. Also, Penguin platform was able to identify the unique genomic location of pseudouridine and the extent of the pseudouridine modification. As a future work, we are looking forward to integrating deep learning models in Penguin platform and improving the performance of random forest model when testing that model with independent cell line. GitHub for Penguin is publicly available at this link. And finally, I would like to thank people in Jungle Lab for continuous support, particularly Alexander Cranon for giving valuable comments on this work, and Ratanon Konchak for some valuable discussions. Thanks for your attention, and I'm ready to get questions. Thank you.